maker. I met Lane when I was in my freshman year of college. She came in to give a presentation about what she was doing during her research experience and her PhD program. And I was so interested about the project that she was working on, and I was so impressed that people can actually do that and make that happen. So I walked up to her and I was like, "Hey, is there any chance that I can work with you?" And she's like, "Sure, let's make it happen." And we've been working together for 5 semesters already. We've worked in several different projects, research, personal programs, projects. There's just been so much growth in the last couple semesters working with Lane. She has a couple slides for her to introduce herself, but I'm just going to do a small intro. She studied biomedical engineering at the University of Arizona. Um, and she just finished her PhD program for biomedical engineering, if I'm not wrong, at the University of Arizona as well. So soon to be doctor. and yeah, hopefully. And she's going to talk to us about her projects and what she's been working on. So Lee, the floor is all yours. Thank you so much for being here with us.
Sam, amazing intro. Thank you. Okay, let me go ahead and share my screen. I had a lot of fun throwing this together. Ended up just going down memory lane a little. Coming to the end of this PhD, these projects have all come pretty far. So what I prepared for you guys was a bit of a complete smorgasbord of random photos from the progress of this journey, what I've learned along the way, and then kind of the open areas where we need more people to come in and put their own touch on this area of research. So my research is in biomedical engineering. That's what my undergrad degree was in, as well as this PhD. But more specifically, it's in the field of biosensors. And even more specifically than that, what my lab really specializes in doing is taking existing technology that's maybe been around for a while, but is expensive, time consuming. You need to have a lot of training to perform the tests. And what we do is we look at these areas that can be improved by maybe making it easier to perform for somebody who doesn't have as much training. A lot of the ways that we do it is just by making it cheaper. For example, I'll go into our smartphone microscope. And so this is what I really love. We live in a world where a lot of stuff has been invented already, but there is a huge need for making these technologies a little bit more accessible so that people in the public can be doing this from your home and just making it a little bit more available to everybody, because this stuff is really fun to do. So what I've got here on the top right is actually a paper I worked on in my undergrad, and I wanted to include it here. It's not my main PhD research. However, it's extremely relevant to the field of accessible tools, because what it is a smartphone imaging device to assess whether your plant is healthy or not. And this is a very interesting project because basically our phones have a flaw. If you talk to somebody who makes the smartphone camera, they'll say it's not very helpful, but it actually, when you're taking photos, you're getting a little bit of near infrared light into that camera. So while what you want to have is just the visible light, which is what we can see with our eyes, some phones actually have a defect where they can see near infrared. But what's cool is that plants, actually, the water in those plants, they respond to near infrared light. And so we were able to figure out this flaw in a smartphone, take advantage of it, and turn it into a tool where you can be imaging your plants and getting a rough idea of how much water they have, or if they need a fresh up. So that's just a brief intro to some of the cool things out there that we can do with a lot of the technology we already have, like your phone. Some of the other stuff that I'll go into is how you can use 3D printing. This is one of the biggest reasons that a lot of this stuff is becoming more available now, because you can just 3D print your own device. And that's really made a lot of this a lot more accessible. And then in general, a lot of my research has also been on lateral flow assays. So in the bottom right there, I just included a little photo of one of those rapid COVID antigen tests. This is exactly what our lab does as well. We're not mass producing it for the public, but we're trying to make it accessible so that people can make these tests on their own. And then maybe you can use it not just for COVID, but for any other virus or bacteria, or maybe even the non-harmful chemicals that you want to look for. Okay, so let's get into the photos. Okay, so for 3D printing, this is awesome. They have some, they're still fairly expensive. You know, I'd love to see these things being free and readily available, but you can buy a pretty basic 3D printer for a couple $100 now, and that'll get you started for being able to make pretty much whatever you want. So we've got a little Darth Vader there. But in the later slides, I'll show you some more examples of the microscopes that we've been building using 3D printing as the case to hold it all together. In the middle there, so point of care, that's another way of saying portable, basically. We want to be able to take this stuff on the go. One of the main reasons that a lot of these technologies aren't accessible is because you have to go into a lab, and not everybody has access to a lab. So everything that we do, we make it so that you can take it with you on the go and do it out in the field or from your house. So right there is a little flow rate video, which Sam was helping me out on. We actually took it outside to see if humidity was affecting our data. It looked like it was going to rain in Tucson, so I thought it was going to be humid, but it, wasn't humid at all. And then the final category of how we can make the stuff accessible is the fact that we do all of our experiments on paper chips. So while some other labs are doing this stuff on PDMS, which is just a organic polymer, it's very beautiful polymer, but it's a little bit expensive and difficult to use. And so paper chips are really exciting. I'm obsessed with them lately. All we do is we get the paper, we stick it into your classic printer, except instead of an ink cartridge, you've replaced it with a wax cartridge. And this just means that you can, at the click of a button, design any sort of test design that you want to try out, and you can print it. All you have to do then is melt the wax, kind of just like melting a candle. And that'll create these nice channels where you can have your liquid flow through. It's really cool. It's very elegant. It's easy to design. And this is a huge area that I think is going to boom in the future for people being able to design their own tests. Okay, so here is one of our smartphone microscope devices. This was actually used for the coronavirus project. It was a big group of people in our lab, and it was a really fun project to work on. And there's a lot of different components. Our main goal is to do the complicated stuff in the background and then make it simple for the users on the other end. But basically, we're using chemicals to find the virus. This is a process called immunoagglutination. I could nerd out about it for hours, but it's basically just using the microscope to find the virus. Then the hardware, we actually need to be able to see it. So that's including the microscope, the 3D printing, the LEDs to light it up, all of those things. And then the software for analyzing. I didn't end up including a lot of software talk in this little mini presentation here, but this is also a huge area that's making all this stuff more accessible now, because anybody can code from their computer or even your phone now. And in the past, when these things were extremely expensive and it was more difficult to get access to computers, it's definitely moving in a promising direction where we can perform very complicated analysis of large amounts of data very quickly and easily. Okay, so 3D printing, if you wanted to make your own microscope, what would you have to do? First of all, this is still in progress. I'm really proud of where it's come, but it's not, don't get too excited. But all you have to do is buy a microscope off of Amazon. You can also make your own using a lens, except that's getting you into the optics. And our whole goal here is to make it simple. So I actually have an example here. This is a cool little microscope. The other one that we have is, can you guys see my pointer? Well, this is one that we use, but this is a different one. This is really cool. You can hook it up to your phone, and then you can see things. And I spent too many hours. I don't know if any of you have seen those YouTube videos where you can get your scalp analyzed. Well, I saw one of those and I was like, oh, I have a microscope. I could do that. And it was gross. So yeah, I recommend doing it fully. It's a really fun experience. And then what else you need is you need your smartphone. That's providing the camera. That's how we're replacing the whole entire microscope just by getting this one little thing from Amazon or wherever else you want to buy it. And then you throw in your smartphone so you can actually collect your images for analysis. And the really cool thing about 3D printing here is that you can make it adapted to whatever microscope you have or whatever phone you have. So it's very easily to adapt and change depending on your needs. We also made a little app down here on the right. It's on MATLAB Mobile. Now, this is a huge area that could continue to be improved. We did this in MATLAB, which is a great program. However, it's mainly, I only had access to it through the PhD program, and it's not very accessible to the public. And so something that I think is a huge area that could be improved in this is just making our own apps through open source software like Python. Okay, so paper. This is very cool too. So here's just a small sample of the thousands of photos on my phone. I have a lot of photos of paper on my phone. And this just is to show you guys that you can really change it depending on what you want to do. So you can see that every single chip, we call them microfluidic chips or micropads. That's just the terminology. It's because paper is very cool because it has liquid pulled through the pores through capillary action, just like a plant will suck water through its stem. And that's a micro size. So that's why it's called microfluidic chips. But you can see how many different designs we were able to try out. So these ones are long and they have multiple inlets. This is just done to control the flow, make it faster or slower. Then you can see down here, we experimented with different levels of wax. Samantha actually did a lot of work on this for us. We were curious about how the amount of wax you're using affects the test. There's basically just a bunch of variables that you can analyze. And this is why we need a lot of people doing it, because every test needs to have thorough analysis and you really need to investigate it to see how this variable affects your flow, how the flow affects these variables, and vice versa. I also wanted to include this middle photo here. This was actually just a fun side project, as many of these things are. You can make your own microfluidic chip using crayons and then melting it. So crayons have wax in them and it melts. And we actually found that it worked decent. Definitely not good enough to get consistent data for a paper published, but this science is very translatable to our everyday lives. And if we continue to understand it better, then we'd be able to start doing these from home. These were, we just drew them out and melted them. And I've also actually done this at home just with my oven. I had a little photo here. So this is just printer paper and I drew the channel and then you stick it in the oven and melt it and you've got a little channel that you can use. A second thing we wanted to look into was whether origami could be included in this. Origami is a very beautiful art. And we were curious if you have two reagents on different sides of paper, if you squish them together, then they will interact together. And this I didn't make it to the final paper either, but there's a tons of different things that you can experiment with, and paper-based microfluidic chips is a very exciting field. Okay, so optical filters. In my research, we use fluorescent particles. These are tiny, tiny little particles that are kind of just like if you get those neon glow sticks, they glow in the dark. This is the same thing, but they're so tiny that you can only see them glowing in the dark if you have a microscope. But the problem is that if you have a bunch of other light, you can't see them. It has to be in the dark. And so what we do is we use optical filters to cut out the light that we don't want and only be looking for the fluorescence that our particles are emitting. However, these filters are extremely expensive. I mean, it makes sense. They have to, it's really interesting, and I still don't understand quite how they make them, but it's glass. They got to get the angles all right. They have to get the coloring in it all right. It's a very, very intricate process. And so, of course, those things are really expensive. Previously, we bought them and they were a couple $100s at least. So something that I was really excited about on this new project is we found out that feeder stage They use color filters, acrylic filters, to change the lighting on set. And those things are actually doing very similar jobs to what our optical filters were doing. So here we've got a UV LED on the top, and then we were trying it out with different filters from this booklet that I think cost $9 off of Amazon again. And so here you can see that the green one didn't work on the left, the one on the right didn't work, but the middle one, it successfully isolated the light that we didn't need, and instead you're just seeing the UV light coming through. And so that's just using, I've actually got the booklet here. I got it for myself to play with. So here, it's just a booklet and it has all of these different colored filters, and you can mess around with them and figure out what you're looking for. Something that would be really interesting to try is to repeat that original smartphone and plant stress project to assess the NIR wavelengths from plants. Maybe one of these filters would work for that. And then additionally, the diffraction coating, okay, here we go. So this photo on the right, this is using a diffraction coating. These are used as well in a lot of microscopes and analysis. However, it's very, very small grading. And so we were actually able to repeat that just by having small slashes in the filters, and it creates this beautiful rainbow effect that's very fun for photo taking. Okay, so here are the fluorescent particles, and this is just to give you an idea of what we were able to achieve with our smartphone microscope. In the middle here, this is a giant clump of those fluorescent particles. So we have to do a lot of processes. The chemical side of this research is still very in-depth, and it would definitely have to still be performed in a lab. But once you finish it, then you can look at it under a microscope. We're still not as beautiful as benchtop microscopes. That's the photo on the right there. It kind of looks like a bit of a starry night. It's very gorgeous. The photos that we're getting with our smartphone microscope that we built can be seen here on the left. So the quality is lower. We have a little bit of less resolution, so we're not seeing as many tiny particles. However, you can definitely see that we're still getting them coming through. And so for our research, where we just need to be looking at the really big clumps, this was perfectly sufficient, and we were able to find coronavirus successfully. So one other thing that I had here to talk about is surface tension. This is a very interesting property in nature that we use in our research. And so I thought it was worth a mention. It's basically, it's why insects can walk on water. If you drop a toothpick into your water, it's why it floats. It's very interesting. But what we actually found out is that if you, let's say, drop a toothpick onto water without any viruses or bacteria, it's going to behave differently than if you drop it into a clean, fresh glass of water. And so what this basically means is that water's properties are going to change depending on what is in that water. It kind of sounds obvious, but for research, then you can really use this knowledge to perform these assays and detect things at very, very, very small levels, where previously you would have had to spend tens of thousands of dollars to find them. Now you can do it just by looking at their properties. So this was a very fun project. Our actual publication is using those paper microfluidic chips and tracking the flow rate, but I wanted to include this because I just think the droplet photos are so beautiful. You can basically just look at how big the droplet gets, and that directly correlates to the surface tension. We had to get a little creative because we only had a very small amount of these clinical samples. And so we had to get a little party hat, have a little capture device underneath to make sure you're not losing any of that potential data. So even in the lab, we have to get a little creative and make do with what we've got to keep it going. And this is really cool here. You can see that on different types of materials. So on the bench, on the 3D printed chip holder, on a plastic slide, and on a glass slide, they all have different properties. So water is also going to change depending on what platform it's on. And that's been a really interesting discovery for this research. So the final thing, we've also been more recently working on a new project for the detection of PFOA. This is called kind of commonly the forever chemicals. They used to be in a lot of water resistant coating, firefighting foam. They definitely are not being used as much now. But there's definitely a lot of interest in detecting them. So just like we need to detect viruses from water samples, we also need to be looking for these other non-viral chemicals that can be dangerous. And something that's really interesting here is this is not at all how the project ended up being completed. We had to take a completely different path. So there's a lot of changes of directions in research, but it was really interesting because you can see the different colors in the water and they're directly corresponding to the amount of PFAS in the water. So even if you aren't actually visualizing using a microscope to see those tiny particulates, you can actually use larger changes such as color to track that motion. And then we repeated this on those paper microfluidic chips again to see how that turned out. So then just a final slide. This was not for research, but this field is very interesting of biosensors. Nature has a ton of things out there that can give you information about your environment, about the water, and so much more. I know I'm only in a tiny little niche, but the big exciting part of this is that I think that we can start to bring this stuff home. So here I was really into nails. I think that was a bit of a coping mechanism with a PhD, but I actually found out that you can paint your nails a certain color and it's basically a replacement for an optical filter. So maybe one day we'll have little carry around filters on our pretty nails to help us analyze plant happiness. And then on the right, I actually, this is a fake nail, don't worry. I stuck a nail through it and then glued a small glass bead to make a little DIY microscope. So maybe we'll also be carrying around our own little microscopes in our pockets. And this is just a fun little nugget of thought that I think in the future we could, just like now we have our smartphones and internet to carry around with us, maybe eventually we'll also have a full lab just in our pocket ready to diagnose and test whatever we want out in the real world. All right, thank you so much.
Thank you so much, Lane. This was such a fun presentation, and getting to hear about how you can use technologies as just 3D printers and like paper to be able to, detect and measure and make all these changes. It was just so great, and also learning about your side projects that you do at home and how about like how using something as our nails we can like create a change within like our environment and use that to measure stuff. That was so fun. These are things that one does not even like think about. So that was such a great presentation. Thank you so much. I do have one question though. So now that you're done and that you've gone through all this like entire discovering process and like developing and learning about these technologies and how they can be used, what advice would you give people whenever they're like frustrated about how pricey STEM can be? Here we are, like we've got you working on your PhD projects and programs, working with paper and just like our phones throughout the entire duration of the program. So like what advice would you give people whenever they're frustrated over that, and how do you get to those like point of care solutions that you've discussed with us?
Oh yeah, that's a great question. I think that the cost of all of these devices is for sure frustrating. And even in my field, which is about making low cost devices, sometimes you'll hear somebody say that and then say that cost a couple $1,000s to make. And I think there really needs to be a lot of education about our research so that we can just continue making it more affordable and actually affordable. Like I think that anybody should be able to have access to this stuff. And the main way that we can be doing that is by making those educational materials to teach about it. And that will just expedite like understanding this stuff more. Like we didn't understand microscopes as well before. Now we do. So we know how to make it a little bit easier, maybe on our nail. But we just need to keep learning about all these other things too now.
Yeah, thank you. And do you have any advice to give younger people or like people that are interested in STEM about like pursuing any STEM paths or STEM careers and stuff like that?
Yes, I say start doing little projects on your own. I think that it took me a while to embrace my my nerdiness. And as soon as I did, that's when things really started getting fun. Once I actually started doing little interest projects on the side is when I actually started understanding classwork better. And I think that all of that personal interest has really translated to a more solid path in career and academia and all that. So whatever you guys think is interesting, I say go for it and do your own little experiments in your kitchen and see what you can learn.
When you said that, a memory came into my mind. I was so young and my mom gave me a microscope for Christmas, I think. And I would go into my patio and just like get leaves and like dead ants or whatever I could find and just look at them through the microscope. And it was like back in the days when internet was like not really a thing. So if I wanted to learn about that, I had to watch like Animal Planet and stuff like that. So that's, I guess, how my interest in biology started. And for sure, like I remember going into classes and like not liking about like physics and optics and stuff like that. But then when I actually got to see like planets or like stars through microscopes, I was like, well, this is actually fun. And that's when things started to get like more and more interesting and fun, and just like seeing real life applications makes everything so much understandable. But yeah, I don't know if anybody.
That is so cute. I love that. I think that every kid should have their own little microscope so they can just stare at stuff, because things are crazy under a microscope.
Like I remember just like whenever I started working with you in the lab, they were like, paper is so fun. And I'm like, it's paper. And then I looked at it through the microscope for the first time and I'm like, it is so cute. And then when I started learning about it, I was like, it has such cool properties. And yeah, totally, like the more into depth that you can literally look into stuff, but like into depth, like literally through a microscope, the more interesting they get. So yeah, I totally agree with that.
Oh, music to my ears. I love to hear that.
But yeah, I don't know if anybody from the audience has any questions, or if you would like to just like give everybody one last set of advice, or just, something you would like to share to close the session.
Well, I peeked at your guys' itinerary and I saw that the next group is talking about failures in entrepreneurship and those lessons. And my immediate thought was, okay, my slide deck is all photos of failures that we had. And I just think it's great to fail as much as you can, because all you're going to do is learn or have a little idea for trying out later. But this session seems really cool, and thanks for inviting me.
Thank you so much for making the time and sharing all of this stuff that you're probably already pretty tired of after looking at your dissertation for like the past month. But thank you for making the time and, talking to us about all the stuff that you've learned. I really appreciate it. We do have like 1 minute before the next session starts, but none of this